Hi everyone. Firstly, thank you all for joining our webinar today. This is the second webinar in our series of 4 condition-specific webinars, which is an extension of our Retina 2020 virtual program. My name is Niamh and I'm the Research Officer of Fighting Blindness. And I'm just going to briefly go over a couple of housekeeping rules or housekeeping notes before we kick off this evening. Firstly, as you'll have noticed, this webinar is hosted over the Zoom webinar function. So during the course of the webinar, you can turn on your cameras or your microphones. But if you have any questions for the speakers, you can put it into the Q&A box, which you'll find on the bottom of your screen. And just to note that you can send these questions in anonymously, and they'll just go directly to our speaker or panelists. And we'll try to get to as many questions as we can, but our agenda is very tightly packed today, but we will get to as many as possible, and we'll do our best to follow up afterwards if not. This webinar will also be recorded, so we'll be in touch in the next couple of days with the link to where you can watch back the recording, or if anyone might have to drop out early. And we'll also have a short survey, a feedback survey, which we'd be very grateful if you could fill out. It should just take a couple of minutes. Yeah, I think that's everything. I'm going to hand over to our CEO of Fighting Blindness, Kevin Whelan, to give the official welcome address. Kevin, if you could just switch on your camera and mic when you're ready, and we can get going.
Sure.
Thank you. Cheers.
Thanks very much.
No worries.
Thanks, Niamh, and thanks for the introduction. On behalf of Fighting Blindness, I'm delighted to invite everybody to this second webinar. And we received very positive feedback from the first webinar in our series of 4. We're particularly indebted to Roche, who are sponsoring this webinar series 2 for us. And also we're very indebted to Professor Jane Farrar, Professor Lorraine Cassidy, Professor Colm O'Brien, Dr. Alison Reynolds, and Dr. Jackie Turner in relation to their contributions to this discussion. And we're particularly pleased that the caliber of presenter is such a high standard, because the information that will be gleaned from this, we feel, will be hugely beneficial to our audience. And it is so important that our membership and the people we represent are kept as informed and up to date as possible in relation to matters that are very particular to the specific condition that they're living with. They or their family member. So for that reason, it's so good to be able to introduce such a caliber speaker. As Niamh said at the outset, we do encourage people to ask questions, and rest assured, whatever we cannot address today, the research team led by Rachel Birmingham and Niamh will respond to each of the queries. We thank you for joining the session, and I am very pleased now to welcome Professor Jane Farrar, School of Genetics and Microbiology, Trinity College, Dublin. And Jane is going to present on the clinical update on LHON and optic neuropathy. Thank you, Jane.
Okay. Apologies there. I thought I'd lost the screen for a second. Okay, so I'm just going to share my screen. First of all, thank you very much for the opportunity to talk today, and welcome everybody. And I'm just going to now share my screen because I have some slides just to share with you. So let me just do that and see if that works well. So could somebody let me know that they can see the screen?
Yeah, I can, Jane. Yeah, looks
okay,
good.
Super. That's great. That's great. Okay, so I was asked today to talk a little bit about the research in neuro-ophthalmology and a little snapshot of kind of the types of research that are ongoing, and also the emerging therapies. And I work in genetics and micro in Trinity. And so I will focus a little bit on really the gene therapies that are emerging for this group of conditions. Just to say that we've worked in the area of ocular genetics for about 30 years, which is a scary thought. And it's involved lots of different people, some of the people talking today as well, and lots of others too. And very importantly, Fighting Blindness, who've really been there from the beginning, funding the research, and working with us in terms of research into ocular disorders in the genetics department, and of course with all the clinical teams around the country as well, with the patient organizations and groups really focused on patient. And these are the kind of outcomes that we've had so far, but we're not going to focus on that so much today. I'm going to focus on moving forward in the area of neuro-ophthalmology. So the focus of today is neuro-ophthalmology, and when we look at our wonderful retina in the back of our eye, we've lots of different layers. It's a diagram on the left-hand side, if you can see it, and there's about 10 layers in the retina, and the layer that we're particularly interested in today is the layer called the retinal ganglion cells and the optic nerve, and then that light energy is hit by photoreceptor cells, and then it's transmitted through those retinal ganglion cells down your optic nerve and into your brain, into the visual cortex, where it's interpreted. And I mean, sometimes when I look at these diagrams myself, or just think about what happens in our eyes, I just think it's pretty phenomenal, the processing that is going on in the eye. And so we're going to look at disorders, neuro-ophthalmological disorders, which involve retinal ganglion cell loss and dysfunction of retinal ganglion cells, or loss of the retinal ganglion cells as well, and also the optic nerve. And so the disease is involving that part of the retina. We're also, of course, interested in other forms of retinal degenerations as well, involve photoreceptor cells. But today's focus is really the retinal ganglion cells and the optic nerve. And I just want to focus on the fact that it's really timely to address these kind of questions and to look at ocular gene therapies, or therapies generally, for this group of conditions. And why is that? And it's because we now know and understand from human genome sequencing really some of the causative genes causing these conditions. That's one really important element. Another really important element is that we can, in cell models, we can model the condition. We can also model it in little mini retinas called retinal organoids, and also in animal models, in mice and in other animal models. So we've got models in which we can test therapies. We've also got really good, safe ways to get genes into the eye. And one of the ways we do that is with little viruses, but we don't always use viruses. But one type of virus is called AAV. The name doesn't really matter, but we know it's well tolerated in the human eye. There have been human clinical trials with good outcomes. And we have the first approved ocular gene therapy, which is not for the retinal ganglion cells and optic nerve. It's actually for photoreceptors, really, and for the RPE, the other side of the retina. It's called Luxturna, and that is my dog there, and I apologize. I'm going to see if I can do something about that in a second. Okay, so apologies for that. If anybody heard that, that was a dog in the background, and I do apologize for that. Okay, so one of the key things that is enabling, as I said just a minute ago, the development of therapies for these conditions is understanding the genetic basis of those conditions. And that's either gene therapies or small molecule drug therapies. If we know the cause of the condition, we can start to make really targeted therapies. And that's where the Human Genome Sequencing Project came into being. And so I just wanted to give a little background before we move on to some of the key outcomes from this kind of research. Just a little background into a number of different areas that are relevant to driving therapies forward. And one is human genome sequencing. The first human genome was sequenced in 2003, and it cost a few $1 billion. Today, if you want to get your human genome sequenced, it costs $500. And so the technology has ginormously improved. It's much cheaper, it's much faster, and we can really start to understand the small genetic changes between you and me that give rise to disease. Genetic information is driving medical advances. It's driving diagnosis of eye diseases. It's driving the development of therapies, whether they're drug or gene therapies. And of course, we saw that in vaccine development recently as well. The more we knew about the the COVID-19 genome, the better we could develop a therapy. And I just want, before we move forward as well, just to remind you, and I'm sure lots of people already know this, but I just wanted to remind you for this group of conditions, it's particularly relevant for this group of conditions. Oh no, this is just annoying. Can somebody mute me just for 1 second and I'll be back. Okay, so what I was saying is, and apologies for that, that there are 3 billion letters of nucleotide DNA code in the nucleus of every cell that we have. And the human genome sequences allows us to sequence that. But I also wanted to highlight that there are also there's DNA also in a part of your cell called your mitochondria. And mitochondria makes energy. It's the energy generator of the cell. It's got 16,500 bases of DNA in there. And so we have our genome, our human genome has our 3 billion letters of code in the nucleus and 16,500 of code in the mitochondria. And that's what's sequenced in the genome. It's also really important to focus a little on mitochondria because for all of the disorders that we're interested in, optic neuropathies, so that's Leber's hereditary optic neuropathy, dominant optic atrophy, and also glaucoma that we're going to talk about today, there's mitochondrial dysfunction. So actually those mitochondria that are in every cell are making energy in every cell. They're making energy, they make about 50 to 60 kilograms of ATP energy every day. That's what your body does. All of your cells are making about 50 kilograms of ATP energy every day. And that's happening in your mitochondria inside every cell. And there's that DNA genome in that mitochondria that's doing that incredible job, just a phenomenal job. And when there's a little mistake in that DNA genome, that's when sometimes the energy that is made is not sufficient and you get disorders, for example, optic nerve disorders. The other thing I wanted to focus in on is that those mitochondria make a beautiful network inside the cell. They're not just little isolated structures in the cell, they make a beautiful network. So in every cell, you've got hundreds or thousands of mitochondria, and they're giving information to each other, and they're making this unbelievable network and sharing information in the cell. You've got mitochondrial DNA in every mitochondria, and it's sharing information. And it's actually, those mitochondria are fusing together and breaking apart all the time right now in every cell of your body. Incredibly, this network is fusing and breaking apart and sharing information. That fusing and breaking apart is very finely controlled. And when it goes wrong, we get disorders of the optic nerve and retinal ganglion cells. Fundamentally, the way our bodies work is called the central dogma in molecular biology. The DNA in your nucleus or in your mitochondria makes a code called RNA that is then processed into the building blocks in your cell, the proteins in your cell. When we have a mistake in our DNA, it gets translated into a mistake in our RNA, and then the protein that is made is not correct. It has slightly wrong building block and it's not working properly. That's called the central dogma of molecular biology, and it's the fundamental basis of genetics. And it's why we can intervene and understand that if we supply a new gene, it should make a new RNA and make the correct protein. So information about our genomes helps us design potent therapeutics, both gene therapies and actually vaccines. You know, the basis of the vaccines that we are all getting now is genetic information. It's actually like a gene therapy. Really, we understand the basis of COVID-19, its genome, and then we can actually supply that part of the genome that makes a spike protein, the RNA that makes a spike protein. And it's the same principle as actually any gene therapy, whether it's a gene therapy for development of a vaccine, or actually whether it's a gene therapy for developing for the development of a therapy for an ocular disorder. So let's look very quickly at some of the genes implicated in optic nerve disorders. So when we're looking at optic nerve disorders, we can think of Leber's hereditary optic neuropathy, dominant optic atrophy, lots of other optic neuropathies, autosomal optic, recessive optic neuropathies, and glaucoma. And using that human genome sequencing technology, we've tracked on many of the causes of these disorders. So we know many of the genes and encoded proteins that are causative of those disorders. So now that we know disease cause, what is the next step? What is the next step? If you understand the cause of disease, you can design a very potent therapy, hopefully targeting the problem associated with that disease. So the next step is to design novel therapies. And when we're designing novel therapies, there are two stages to it. There's a preclinical stage and a clinical stage. Preclinical is before you go into patients, and clinical is when you go and test in patients. And when we think about gene therapy, gene-based therapies, they can be either what we call in vivo or ex vivo. They can be injected into the patient. So that's where the gene that's missing, for example, that's not working properly, that has a mistake in it, is provided directly to the patient. In the case of an eye disease, you'd be injecting into the eye, or ex vivo, where you actually take some patient cells and modify them. Okay, and when we're thinking about gene therapy strategies, we can provide, for example, the normal gene into the cell, or we can provide a gene with a new function of some sort. So we can actually improve the cell in some way. So there are lots of different gene therapy strategies where we're either providing the normal gene, or we might actually be providing a gene, for example, that helps to keep the cell alive for longer. Or we can suppress expression of the mutant gene, or we can correct the mistakes in DNA itself. So those are the kind of components that we have in a gene therapy. We have a virus that delivers the gene. It could be a normal gene because the patient is missing it. It's injected directly into the eye. It hopefully infects those cells in the back of the eye, in this case, the retinal ganglion cells, and the optimal protein is produced. As I said before, it's important that we have models in which we can test these therapies. And incredibly, they're either cell models or animal models, but also we have retinal organoids. We can take cells from a patient, make them back into stem cells, and then grow a mini retina from that patient. So we have a patient-derived mini retina, and we can test treating that patient's disease in that mini retina. Incredible technology. So given we have all of these technologies, what is the status quo for neuro-ophthalmology and gene therapy? And we're going to look at a couple of examples. Let's look at Leber's hereditary optic neuropathy. It's a complex 1 deficiency disorder. It has mutations in that mitochondrial DNA. And so that mitochondria is not making enough energy. And so if we know which mutations are present, maybe we can supply the normal gene. And that's exactly what people are trying to do. 70% of LHON is due to mutations in the ND4 gene in mitochondria. That's involved in generating ATP, generating cellular energy. And so there are various therapeutic options for LHON. One is deliver the normal gene back. But then there are other options, and I'm sure that, that Lorraine Cassidy will be talking about some of these. For example, there's a drug called idebenone, and I'm not going to focus on it because Lorraine will talk on it. Or we can actually try and keep the cells alive longer, or we can use antioxidants as well. So there are lots of therapeutic strategies, but I'm going to focus a little bit on the gene therapy, where you're providing that normal gene back into the target cell type by injecting the gene therapy into the vitreous of the eye. And notably, there's a therapy in development for the most common form of LHON ND4 by GenSight Biologics. And in that, they have shown in a press release yesterday, June 1st, that there's significant benefit in the patients that are treated by comparisons with patients that are not treated. So the controls, the natural history of the disease that is not treated is compared to the patients that are treated with this therapy. Amazingly, this therapy, when you treat one eye, both eyes benefit. there's crossover, it would seem, of the therapy from one eye to the other down the optic nerve. But clearly, it's very exciting. Their clinical data in human patients is showing real benefit. And that's, as I said, a press release from the 1st of June, yesterday. But what about the other forms of LHON? You know, what else, you know, because it's not just ND4, there are other genes involved as well. And so there are gene-specific therapies that people are contemplating for the other forms, ND1 and ND6, etc. But there are other therapies in development, they're in research development, so they're not in human patients yet, where they're looking at, for example, providing better mitochondrial function, or preventing the cells from dying by apoptosis, or providing neurotrophic factors. These are all gene therapies, but they're overarching. They're not targeting a specific gene. They're actually an overarching therapeutic approach that's modulating the mitochondrial dysfunction that's symbolic of Leber's hereditary optic neuropathy. And this is just a schematic of what's been done. So far, these have been tested in mice, and there's been benefit found with a number of these therapeutic strategies by providing more mitochondrial function or preventing cells dying, preventing programmed cell death. And as I said, there is also a drug therapy approved for LHON from Santhera, and that's idebenone or Raxone, and I'm sure that Lorraine will be talking to you about that. The second example is dominant optic atrophy. In dominant optic atrophy, about 1 in 25,000 people worldwide have it, whereas 1 in 10,000 people in Denmark have it. It's very common in Denmark by comparison with the rest of the world. The primary driver is a gene called OPA1. Again, it's important in the mitochondria. It's actually involved in doing that, breaking apart and closing together the network of the mitochondria, the fission and fusion of mitochondria. That gene and encoded protein is involved in controlling that process in mitochondria. And it's involved in about 80 to 90% of cases of dominant optic atrophy. There are no gene therapies actually in human patients yet, but there are lots in animal models and in cell models where people are testing therapeutic strategies for dominant optic atrophy. For example, there's an OPA1 gene replacement strategy that is being tested for dominant optic atrophy. That's providing the normal gene back into the target tissue. And that has been found to provide benefit in mice. So providing that normal OPA gene back into the target tissue helps mice with dominant optic atrophy see better. However, for any gene therapy, we have to be very careful, because actually what's really important is getting the right level of the gene therapy. Too much of a good thing is bad. And that's a really careful consideration that we have to take into account right now for dominant optic atrophy. We don't want to overexpress that gene. We want to control it really, really carefully. That's the same for every gene therapy. We got to get the right level, the right dose of the therapy. The last thing I'm going to talk about, but only very briefly, is glaucoma. And that's because I know that Colm O'Brien will be talking to you a lot about glaucoma. But the therapeutic strategies we've been talking about, the gene-based kind of therapies, they're also applicable to glaucoma, which is a very common disease. It's predicted by 2040, about 110 million people will suffer from glaucoma. This involves a vulnerability, again, of the optic nerve head and the optic nerve, and loss of those retinal ganglion cells in the retina. And there are lots of people thinking of ways of treating glaucoma with gene-based medicines, gene-based therapies. There are different types of glaucoma: primary open angle glaucoma, primary closed angle glaucoma, and there are many different factors involved. I showed you in one of the first slides lots of genes that are risk genes for glaucoma, but there are lots of other things like intraocular pressure, many other risk factors, age for sure. It's an aging disorder. It's more frequent as we age. And the key thing is the buildup of pressure due to aqueous humor that doesn't flow out appropriately. So we have a buildup of pressure in the eye that results in retinal ganglion cell loss and optic nerve loss in glaucoma. As I said, there's lots of research, lots of work in cell models, in animal models of disease, of therapeutic approaches for glaucoma. One of them is disrupting a gene that's involved in making that aqueous humor. If we make less aqueous humor, we'll have less pressure. And when we have less pressure, we won't lose our retinal ganglion cells and optic nerve. That's the idea. And there's been benefit with that therapeutic approach in 2 mouse models of glaucoma. Another idea is actually to use to try and open the junctions between cells. So actually the aqueous humor flows out more easily, and therefore there isn't a buildup of pressure. And that's shown benefit in 1 mouse model of glaucoma. And in fact, Colm O'Brien was involved in that work. And also, there are lots of therapeutic strategies where people are just trying to keep those cells alive, the retinal ganglion cells in the back of the eye, alive with neurotrophic factors. This is just 3 examples of literally tens of examples of therapies that are being tested, but in mice and in animal models and cell models for glaucoma. So just to summarize, there are many therapeutic strategies for optic neuropathies. Many are gene therapies, some are drug therapies. People are looking at correcting the primary defect, for example, ND4 gene therapy or OPA1 gene therapy, but other people are looking at modulating, adding mitochondrial function or protecting cells from dying with anti-apoptotics. So lots of therapeutic strategies. Much of that is still in where we call research. There are some in clinical trials, but much is still in research. So just to summarize, we are in the era of genomic medicine. Genetic information is driving medicine. It's driving our ability to understand the basis of disease and therefore to design potent targeted therapies. There are many genes already identified for optic neuropathies, for Leber's hereditary optic neuropathy, for dominant optic atrophy, for glaucoma. So they're targets for therapies. We see that ND4 is looking really good for LHON. We see OPA, not in clinical trial in humans, but in mice, is providing benefit in animal models of disease. So the gene therapies that are in these studies, in preclinical, that's before humans, or clinical studies, are either targeting the primary defect or modulating the disease course in some way. We have a truly exciting decade of gene therapy ahead, for sure. So I just want to acknowledge lots of the teams that I work with here, lots of the funders that fund our own research, even though I wasn't really focusing much on our own research. And of course, the many national and international collaborators we have. And again, to emphasize the really important role of fighting blindness in driving this research forward in our own lab and throughout Ireland, in really understanding the genetic basis of retinal degenerations, optic neuropathies in Irish patients, and in helping to design therapeutic strategies. So thank you. And I'm really apologize about my dogs. Sorry about that. Okay, so
thanks very much, Jane. That was brilliant. And don't worry about the dog. I think we all understand. I think in the interest of time, we won't take any questions, but I think there was a few that you covered really well throughout the talk. And as I mentioned, we can follow up if anyone has any questions that might come in after. They can email the research department. But thanks again, Jane. That was brilliant. And we'd like to move on to the next speaker this evening is Professor Lorraine Cassidy. Lorraine is based in the Royal Victoria Eye and Ear Hospital and specialises in neuro ophthalmology. And she's going to speak to us today about some of the clinical updates associated with the conditions today. Lorraine, if you could just turn your camera and your microphone on and maybe share your screen when you're ready. And there we go.
I've unmuted.
Oh yeah, there we go.
Can you hear me now?
Yeah, we can hear you.
Okay, that's fine. Actually,
Yeah.
I'm going to just show so that I can use my pointer. I'm not going to have, I might just have quite small slides. So I might just stop the video and just share the screen. Here we go. Can you see my screen now?
Not yet. No.
Okay. So I've put share screen, and
Yeah, we can
can you see it now?
Yeah, we can. Yep.
Lovely.
Perfect.
Thank you. So
Thanks.
good afternoon,
Thanks,
everybody, and welcome. My name is Lorraine Cassidy, and I'm an ophthalmologist in the Eye and Ear Hospital, and I also work in Tala Hospital. And one of my special areas of interest is Leber's hereditary optic neuropathy. And today, I would like to give you an update on the treatments available and those that are in the pipeline for Leber's hereditary optic neuropathy. Now, a lot of what I'm going to say, Jane has said much more eloquently and in much more detail. And before I start, what I'd like to do is to give you, in order to help you to understand the different treatments, I want to give you a very simple overview of how the visual system works, and then to kind of talk about what's gone wrong in Leber's hereditary optic neuropathy. So when we look at, for example, at a person, if we look, for example, at a person or a person's face, that image is focused by the eye, which is a very complex camera sitting in your skull. It is focused on the retina here. That is converted into an electrical signal, which is then transferred up the optic nerve all the way to the brain, where it's interpreted in the back of the brain called the occipital lobe. And then this is passed forward into the occipital lobe, or the frontal lobe, where your personality lives, and your brain understands that you have seen something. When we look inside the eye and look at the retina here, this is what we see. The image projected, so for example, if you're looking at a face, the image is presented right here on the retina, particularly the details of the face, i.e. the color of the person's eyes, their facial expression, their lips, their nose, is focused on this very highly specialized area here called the fovea, which is the part of the eye that job is to decipher very, very fine detail. And this is made up of the very thin layer of ganglion cells that Jane was talking about. And then the details of the image are passed along from the ganglion cell via what's called the papillomacular bundle to the optic nerve, and then it travels along the nerve to the brain. This is a diagrammatic scheme of what's called the papillomacular bundle. And for the eye to be able to transmit the image from this fovea here, from the ganglion cells, along the papillomacular bundle to the optic nerve and up to the brain, this requires an enormous amount of energy. And Jane has already touched on this in much more detail, but I'll just give you a very simplified version of what Jane has said. So how is this energy actually produced? Well, the energy is produced in the ganglion cells here the same way it is in every other single cell in your body. So I'd like you to think of your body as being composed of trillions and trillions of cells. And in each cell, there are these tiny little organelles called mitochondria. And these are the powerhouse of the cell, or I say like the boiler in your house. And what they do is they convert the food and the oxygen that you consume into energy that allows each cell in your body to function, no matter what that cell is. If it's a kidney cell, a brain cell, a muscle cell, or a cell in your eye. In Leber's hereditary optic neuropathy, what has gone wrong? Well, the DNA that codes for making the mitochondria, as Jane has already told you, is defective, and it makes an abnormal mitochondria, and also it makes the mitochondria very inefficient because it cannot convert the food and oxygen into energy efficiently. This in Leber's only affects the mitochondria in the ganglion cells and in the papillomacular bundle. Hence, the detail of the image cannot be transmitted to the brain because there isn't enough energy to do this. Because you don't have enough energy and you start to produce these byproducts called antioxidants, what are oxidants? What happens is you get oxidative damage to the tissues and the ganglion cells begin to die. And this is when somebody starts to present with their vision, their central vision going down. And usually the patients present with loss of vision in one eye, and then a few weeks later, the second eye goes and you lose your central vision. Here is a scan. We're able to look at that layer here, and we're actually able to measure the thickness of the ganglion cell, thanks to all these very clever scientists who developed these amazing machines. This is called an OCT machine, and many of you patients who come to the clinic will have an OCT done up in the photography unit where we're looking at lots of different things. Professor O'Brien will be looking at your optic nerve to see how badly the glaucoma or how little the glaucoma has affected your optic nerve. And what I'm looking at in patients with Leber's hereditary optic neuropathy is I'm measuring the ganglion cell thickness. I can't measure how the mitochondria are functioning, but I can measure the thickness. And here is a normal person with normal thickness ganglion cell, and that's usually in and around 75 to 80 micrometers. We measure it in micrometers. Here is a patient with Leber's hereditary optic neuropathy whose ganglion cell has been damaged. This is the early phase of the disease. They still have quite a lot of ganglion cells, but the thickness has gone down from the normal 75 to 80 to around 50, and this can be much less. So the central vision in these patients is affected. So I just want to step back a little bit now and just show you again. I'm going back to the mitochondria, and I want to talk just to help you understand how the drugs work. How do the mitochondria actually make energy? How do they convert food and oxygen into energy? This is a horrible word that us doctors and scientists as young students had to learn, this awful thing called oxidative phosphorylation. And this is a very highly complex chemical chain reaction which occurs in the mitochondria and allows the mitochondria to convert food and oxygen into energy to allow the food to work. And in Leber's hereditary optic neuropathy, this chemical reaction is interrupted because the first protein in the chain is broken. And when this is broken, if the oxygen and the food cannot get in here, then it can't go along the rest of the chemical reaction and produce energy. Hence, you don't have any usable energy in these abnormal mitochondria. So this is in essence what it's all about. How can we interfere, or how can we help the defective mitochondria in the remaining ganglion cells? Because if you have Leber's, you will still have remaining ganglion cells. They're just abnormal and they're not working efficiently. So how can we help these cells to produce more energy and to improve your vision? Well, the first thing we can do is we can bypass the faulty protein in the chemical chain, or we can try, as Jane has talked about already, to change the faulty DNA by making it into normal DNA and try and produce normal mitochondria. So idebenone works, or Raxone as it's also known as, works the first way. What it does is it allows the oxygen and the food that you've consumed to bypass the first reaction in the chain, if you like. So it allows it to go on to the next one, and it allows it to mosey along the chain and produce oxygen or produce energy, and hence allow the visual ganglion cells to function better. This varies, of course, amongst individuals on how it improves your vision. And I've been treating patients, idebenone has been available for the last 2 years in this country for patients who've had Leber's for less than a year. And in my patients, I find that I've had 1 or 2 patients who've had very, very good results. I've only had 1 patient out of about 20 who's had an astounding result and has been able to drive again. Again, I don't know if it's due to the idebenone or whether it's due to the fact that he's had a spontaneous recovery, which is something that can happen. Then I've had other patients who, when we measure their vision, we measure their visual fields, we measure their ganglion cell thickness, they've had absolutely no change. However, they tell me that their quality of life has changed enormously since they've started the treatment, and that they can see things that they couldn't see before, and they can function better. They can't really explain it, but they say that their visual function is better than it was before. The other way that we can alter the production of energy in our ganglion cells, Jane has already told you in a lot of detail, is to alter the DNA. And this is where Gensight's new treatment, the Lumivoc, has come in. And this is where we inject a normal DNA into the vitreous. This then is transferred into the cells in the ganglion cell layer containing the abnormal or mutated DNA. And then this will hopefully be imported into the mitochondrial DNA, resulting in the production of normal mitochondria and normal energy, hence allowing you to see again. And there have been lots of very clever people who have done some amazing studies. And those of you who are listening today who have Lebers will, of course, be desperately looking up all the time what studies are being done, what is there coming online that's going to help you. The RODOS study is a study that was published in Brain in 2011, and that was a study looking at idebenone. And those of you who are able to have idebenone treatment have come to me in the eye and ear already and are already on your idebenone. Then we have, of course, these very exciting genetic trials coming up. So the REVERSE and the RESCUE study, the REVEAL study and the REFLECT study, these are all trials that they're very exciting. They're phase 3 trials looking at actual people with Leber's hereditary optic neuropathy where the correct gene has been injected into the eye. And as Jane has said already, they have found that not only did the injected eye have a result, but the second eye in some of these patients also had improvement. Now, the improvement is small and it's very individual, but it's at an early stage, and we are hoping that soon that we will be able to have this treatment available. So what options are available at the end of the day for patients with Leber's hereditary optic neuropathy? At the moment, all we have is idebenone or Raxone, and this is a very expensive treatment, but we are allowed to prescribe it for patients who've had Leber's for less than 1 year, as the evidence doesn't really support any improvement yet in patients who've had it for longer than that. Though I have to say, you know, if you just fall outside the year, I'm still happy to give it. This is available for patients with any mutation. It doesn't matter which mutation, whether it's the 11778 or the 1448 or the 346 mutation. This is available for everyone. In the near future, we hope to have the Lumivoc. Unfortunately, this only really works for patients with the ND41778 mutation. But in the distant future, I'm hoping that all our very clever scientists will be able to genetically modify the other genes. If there are any questions that I can answer at any stage about any of these trials, because they're very, very intense and very well-structured trials, and they're very difficult to interpret. And if anyone wants to email me through Neve, I'd be very happy to answer any of your questions. So thank you very much. That's all I have to say, really. I wanted to keep it very simple. And thank you very much for your attention. Thank you.
Thank you very much, Lorraine. That was really informative. We had a question come in if there was any tablets or treatments coming down the line that could help slow down Leber's, but I think you covered that. And as you mentioned, if anyone wants to follow up with me a bit more specifically, and I'll be speaking to Lorraine, they're more than welcome to email the research department directly. So thanks again, Lorraine. So I'd like to welcome our next speaker is Professor Colm O'Brien. Colm is a consultant ophthalmologist surgeon in the Mater Hospital, and he specialises in glaucoma. So we're delighted to have Colm here today to give us a clinical update on glaucoma. If you want to share your screen whenever you're ready, Colm. And just a reminder that if anyone does have questions, to pop them into the Q&A box, and they can be anonymous. Thank you very much. Thanks, Colm.
Hi, Niamh, is that on screen?
Yeah, that's perfect.
Yeah, thanks very much. Thanks, Niamh, and hello to everybody on the webinar. I'd like to thank, obviously, Fighting Blindness for inviting me to give this talk about the clinical features of glaucoma. I've been looking after glaucoma patients at the Mater Hospital for the last 22 years. And while Jane and Lorraine have given you brilliant talks there about exciting new gene therapies that are actually in clinic or about to come to clinic, I'm going to concentrate in my talk on trying to address many of the worries and questions that I have heard from patients over the last 20 odd years. So that's where I'll be coming from with this talk. And so there's some key messages that I'd like to emphasize at the start. It's a very common condition. So 2% of the population over the age of 40, and that rises to about 5% of people at the age of 80. So that's 1 in 20 of 80-year-olds will have this condition. By and large, patients have no symptoms in the early stages, but later on, it can affect the quality of life if it's undetected and untreated. For most of us, for most people diagnosed with glaucoma, it's detected initially when you go and see your optician to get your eyes tested for your glasses, and then you'll be referred on to your local eye department. And by and large, treatment works. The aim of the treatment is to lower pressure, and the great majority of patients do very well just using eye drops alone. So as has been mentioned earlier by Jane and by Lorraine, it's a disease where the optic nerve at the back of the eye is damaged. Now, initially it affects your side vision, so you tend not to notice it. You also tend not to notice it because your other eye will protect, and there's a crossover between the two eyes. So you may have a degree of disease in one eye, but you won't be aware of it because we tend to function most of the time with both our eyes open. Now, it's mostly caused by having a fluid pressure in the eye that's just too high for the eye. And the high pressure damages the nerve. But we also recognize that having a bad circulation to the eye, or in particular a bad circulation to the optic nerve, will also contribute to the disease. So you've seen this picture earlier from both Jane and Lorraine, but this is where the damage occurs back here at the optic nerve, which connects your eyeball to your brain. The cause of the high pressure is due to a block in the drainage system, which is actually at the front of the eye. But the damage, the end result is damage to the optic nerve, and so that you lose your sight vision. Just to reiterate, I think what Lorraine said, this is the central part of your eye, the one that the part of the retina that you use for fine detail, for reading and recognizing faces. But the message, everything that you see that occurs in the retina, all gets transferred up the optic nerve and up to the brain. So that's where we concentrate our clinical examination. And for most of us, we start off in life as youngsters with over 1 million nerve fibers. So it's like a fiber optic cable that we have. And we have about 1 million when we start off. And as we get older, the nerve fibers gradually drop out. The trouble with glaucoma is that the rate at which the nerve fibers drop out accelerates. So the nerve fibers are visible here in this picture as these white straight lines that are coming out of the optic nerve. And that's what a normal picture looks like. And I'll now show you an optic nerve where the nerve fibers have been lost. And you can't see any nerve fibers in this picture. They've been lost completely from, the nerve fibers have been lost altogether. And this is an advanced stage of disease. So you go from having normal to a gradual loss of nerve fibers. And then with time, if it's left untreated, unfortunately, this is the end result. And when we're examining people clinically, we're looking at a clinical feature called the cup to disc ratio. So the cup that we talk about is the pale area in the middle of the optic nerve. This is the optic nerve in a normal eye. And the pale area has no nerve fibers. These 1 million nerve fibers are actually living on the outside of the optic nerve around here. And so when you lose nerve fibers due to glaucoma. This cup, the pale cup in the middle, gets bigger, as shown here on the picture on the right. And we're able to measure the ratio of the size of this pale area to the size of the optic nerve. In a normal eye, called the cup, they drop down. It's like falling off the edge of a cliff. The nerve fibers just disappear. So that's the clinical picture. And as Lorraine said there, we're now using high resolution cameras and imaging systems, including OCTs, that enable us to monitor and measure the amount of nerve fibers. And what the camera does is take a picture, does a 3D reconstruction of your optic nerve, compares your result with a group of people of your own age for a good comparison. And if you fall outside the normal range, then that's flagged as being suspicious or indeed glaucoma. So we have a lot of different cameras available to us now. And what's interesting as well is that you'll find when you go to see your optician nowadays, many of the opticians now have these imaging cameras, and that's giving us more important information when the patients are being referred on. So to reiterate there, the clinical features that we concentrate on are the amount of optic nerve cupping and the paleness of the nerve, but also we look at the peripheral visual field. And I'll come to that now again shortly. In our clinics, by and large in Ireland, about 80% of patients that we see have elevated eye pressure, high eye pressure. On the other hand, 20% of our clinic patients have eye pressures in the normal range, which makes us think what's the cause of the damage there. And in terms of categorizing the disease, 98% have what we call an open angle glaucoma. There is a rarer, uncommon type, but it presents much more abruptly with a painful red sore eye. This is called acute angle closure glaucoma, but it's not that common in Ireland, but it does occur. And indeed, the other comment to pass here is the fact that by and large, most patients simply have glaucoma. We call it primary glaucoma. It affects the eye and nothing else. On the other hand, a small proportion get glaucoma as a result of having another eye disease, perhaps inflammation or uveitis at the front of the eye, maybe other medical conditions associated with diabetes, etc. Or indeed some medicines might contribute to causing glaucoma. And what we worry about here are people who take steroids for a variety of different conditions which they have to take, including respiratory problems and kidney transplantation, etc. So these patients are at risk of glaucoma if they're on systemic or steroids for a while. So the eye pressure, and that's the key thing in the whole talk tonight. We call it the intraocular pressure, or IOP, and there is a range of normal values. It goes from 11 up to 21. There's a range of normal values for your blood pressure, for your heart rate, for your body weight, for your blood sugar. So we need to know what the normal values are. And once it goes above 21, then we become quite worried about the possibility of somebody getting glaucoma. And the level of pressure is a balance between the amount of fluid that's produced every day in the eye, and then that should be balanced by the same amount of fluid that leaves the eye through a very small drain in the side wall of the eye. Now, that has nothing to do with your tears or anything. It's just a very small drain on the side wall of the eye. We know that our eye pressures are higher in the morning, in the same way that blood pressure is higher in the morning, lower in the afternoon, and lower in the evening. And as we get older, our eye pressure, like our blood pressure, will go up. This is because there is a narrowing of the drain, and the fluid can't escape, and so the fluid accumulates, and the pressure can build up. So that if the eye pressure is too high for too long, this will eventually cause glaucoma. And what I have here is just two pictures to show. Let's maybe look at the one on the right. The fluid in the eye is produced by the structure near the lens, and it's secreted here into the front of the eye, and it drifts with convection currents actually towards the front, and then out through this structure here called the trabecular meshwork of the drain. It's like a sieve, And as we age, the sieve gets blocked with dead cells and debris cellular. And so if once the sieve gets blocked, the fluid can't escape and the pressure builds up. How do we measure the pressure in clinic? For anybody who's been up to an eye clinic, you'll have your eye pressure measured with this instrument here that we place on the microscope. And what's interesting, of course, is that this is a picture to show this prism, which is actually placed on the front of the eye after we put anesthetic drops on the eye. And we're able to measure the pressure within the eye to a very accurate level. And that's routine and standard technique for all clinical examinations. So how do we examine for and monitor for visual field loss? We use machines called perimeters, and there's the most common one that we would have in Ireland is called a Humphrey perimeter. Patients will get their visual field test carried out probably once a year, and it's a difficult test to do. It requires a lot of concentration. It's not easy to do, except that it is very sensitive at detecting early disease. Takes about 10 minutes per eye, so it's a tough test. It's hard to concentrate for that length of time for any of us. But for those of you who've been up to the eye clinic, you'll recognize that instrument for what it is. And there are a variety of different types of glaucomatous visual field loss. This is what we call a nasal step. If you're looking straight at this, and this is on the side of your nose, this is this dark area is the area that I'm referring to, early glaucoma. And if that is your only damage, you won't detect it because your other eye will be able to compensate for it. So that's one of the issues for detection of early disease. Other examples, an arcuate scotoma. There are a variety of different names that we've designed with this. So this one is called a paracentral. It's close to the center of your vision. These dark spots here represent early visual loss in this particular eye. And the thing about this condition is that the visual loss is silent. It creeps up on people. It's slowly progressive. And the trouble is that it's irreversible. So early detection is the key. Unfortunately, patients can go on to lose all the visual field, but yet might be able to see quite well down through a narrow tunnel of vision straight ahead, but lose all the side vision. And how does this affect people? Well, in their daily lives, it's going to cause problems, obviously, with driving, but there's a tendency to have a higher number of falls and fractures. People find it hard to judge steps and curbs, and they have problems going downstairs rather than upstairs, trying to judge where that next step is. Difficulty at adapting, going from a bright room to a dark room. Complain of glare if it's a sunny day, but if it's a dull day, they also complain that they can't see well. So it's a kind of a halfway house for a glaucoma patient. Is a bit of an overcast day. It seems to be the best kind of condition for having relatively good vision. And then obviously having trouble seeing in dark places, which is a similar kind of thing. Now, just a brief few words here on a condition that I mentioned earlier, people with glaucoma who have normal pressure. And about 20% of all the patients in the clinic have nerve damage and loss of vision, but have normal eye pressure. And you could ask, why do they get glaucoma? And there are a variety of different factors here. One is you may well have had a pressure of 10 all your life, and it's gone up to 18. It's still normal, but it's nearly gone up 100%. And it may be that you're just vulnerable to that slight rise in pressure. It's not high, it's not elevated, but it's too high for your particular eye. So you may be that some eyes, even with normal pressure, are sensitive. But there are one or two kind of clinical groups that fit the bill. I won't go through this, it's too complex. But there's two kind of groups of patients that I see with this normal pressure glaucoma. So the first one is the younger group, and then there's the older group. And the younger one, predominantly women, postmenopausal women who are short-sighted, have a history of migraine and poor circulation in the hands and feet, tend to have low blood pressure. They're often very thin. And funny enough, I'll often ask about whether they like dark chocolate. It seems to be a collection of features that often go together. And by the way, you may have all of these things, but you won't, and the majority of people with all these features won't get glaucoma, but it's just some of our normal pressure glaucoma patients are in this group. And then there's an older subgroup of this type of glaucoma, where you have an equal number of male and female, but they tend to have cardiovascular diseases. They have high blood pressure or diabetes, angina, claudication, or maybe even have had TIAs and take a number of cardiac medications. So that's another cohort of people. And the common denominator here to both of these groups is that the circulation in the optic nerve seems to be vulnerable, even to normal levels of eye pressure. So that's a feature of this group. Here's an example of a patient presented in the right eye here with a pressure of 14, and this is the optic nerve. It looks quite healthy. And here's the left eye. Even though the pressure is normal, it's higher in the left eye, it's 18. And this patient has in the left eye an increase in the amount of paleness, an increase in the cupping, comparing it with the other eye. And then when we did the visual field test, it shows some damage in the left eye, but no damage in the right eye. So this, even though the pressure is normal, the higher the eye with the higher pressure, the left eye in this case, has more optic nerve damage and has visual field loss. So the key message with this condition is, although the pressure is normal, treatment is still necessary. And one other subtype of glaucoma that's worth of mention here, because it's common or relatively common in Ireland. It's a form of secondary glaucoma that we call pseudoexfoliation. We have a lot of it here, and it's because of the Vikings. And all the ports around Ireland where the Vikings left their DNA behind, including Limerick, Waterford, Cork, Dublin, we see a lot of pseudoexfoliation. I worked in the UK for 15 years and I saw very little of it over there because the Vikings didn't make a big impact in the UK. Interestingly, they've identified a gene that seems to be, that's a gene mutation that occurs in 100% of patients with this condition. Now, the tough thing about this disease is that the patients tend to have higher eye pressure with more severe disease, and they're often harder to treat. And many of them require surgery to control their eye pressure. And we feel in our clinic, when we audit the results, about 10% of the patients that we see in the matter will have this particular type of glaucoma. So I've just talked about 2 or 3 different subtypes of glaucoma. And now to treatment. Everything we do in clinic is aimed to lower the eye pressure, because we know that when the pressure inside the eye is higher than it should be, it damages the optic nerve at the back of the eye. So all our focus currently is aimed at lowering the eye pressure. I think listening to the developments that Jane and Lorraine were talking about, we may well in the future go on to use and utilize gene therapy, perhaps to improve the function of the mitochondria, not just in the optic nerve, but in the retina and other parts of the eye in patients with glaucoma. So those studies will be carried out, I think, in the next few years, and will probably lead to direct benefits to patients. So what we do with the treatment is to reduce the level of eye pressure by 33%. We know from all the trials that if we lower pressure by this percentage from the initial pressure, then we have every chance to preserve the visual field. Nearly all the treatment is aimed to reduce the amount of fluid we produce, or indeed increase the outflow of fluid in the eye. And the great majority of patients are well treated with eye drops alone. Some require tablets in the short term, and a small proportion of patients go on to require laser and surgery. But the great majority of patients do very well with eye drops alone. And in the last 20 years, we have had a number of new eye drops come to the market and are available now for patients. And so, they're different, what we call first line drops, second line, and then others that we rarely use anymore. And most people who develop glaucoma will start on what we call the prostaglandin analogs, and they're very effective. They only have to be used once a day. About 20% of people get long dark eyelashes and a brown coloration of the iris. So cosmetically, actually, the patients quite like that change. But anyway, so I'm not going to talk too much about the different kinds of treatment. Here's a clip from a magazine that I saw a number of years ago, where in America, a number of states have approved the use of cannabis for treating glaucoma. Interestingly, cannabis has been shown to reduce eye pressure when people smoke hash or And so the pharmaceutical companies have been working very hard to develop an eye drop that behaves like the inhaled cannabis. The trouble is it's very hard to get an eye drop that only works in the eye and doesn't cause any of the systemic or non-ocular issues. So coming towards the end, I'm just going to reiterate my first slide. This is a common condition. It affects 5% of the population at 80 years of age and older. And that's a growing part of our society. It doesn't produce any symptoms early on, but later on it can seriously and adversely affect the quality of life. You know, for example, if you lose your driving license because you've lost your field of vision, then that can be a really significant detrimental effect on the quality of your general day-to-day life. By and large, most patients are initially seen and detected by your local optician, and then you'll be referred to your local hospital. Treatment works. Here the principle is simple enough. The lower the eye pressure, the better the outcome. And by and large, most patients are very well treated with eye drops alone. So I think that's my last slide. Yes, it is. So I'm happy to take any questions.
Sorry, thanks very much, Colm. That was very informative. We have a couple of questions, and I think we have time for a few that came in. One of them was regarding some of the eye drop treatments for the use of glaucoma, and that do they lead to the skin under the eye becoming marked or stained? And if so, is there any tips to avoid that? Do you know? Yeah,
some patients with the prostaglandin analogs, the one that change they make the eyelashes longer and darker, and they can also make if you have a bit of brown in your pupil, then it'll go browner. They can also cause a kind of a purplish discoloration in some eyes, but not all people. So it's difficult to know why. And one way to try to avoid that would be to, when you put your eye drop in, is to simply place your finger over the side wall of the nose, and that seems to stop it from spreading to the skin. But even doing that doesn't guarantee that you don't get this kind of discoloration.
Yeah. Okay, thanks. And somebody else has asked what the difference is between uveitis and glaucoma.
Uveitis is inflammation, and for the great majority of patients, we never find a cause for it. But if you get recurrent uveitis, you're likely to get secondary glaucoma. It's a cause of glaucoma, but it's a very uncommon cause of glaucoma. It probably affects less than 1% of glaucoma patients. The trouble is it's difficult to manage because you have two conditions. You have the uveitis and you've got the glaucoma to control. So it can be a more challenging clinical challenge for us.
Thanks. And just a question has come in by the Q&A box. I think you might be able to see it there yourself, but somebody's asked, have your NT glaucoma patients been tested for Leber's? Migraine and poor peripheral circulation are often reported in LHON patients, and that it was mentioned that there were several NT glaucoma patients that actually had LHON. Yeah,
that's a great question. And we recognize that many of these patients with glaucoma have mitochondrial dysfunction that you've heard about today from Jane and Lorraine. And their mitochondria are not producing enough energy to protect the cells in the retina and in the optic nerve. So yes, there is a bit of overlap here. The one big difference clinically, when you compare the two, that we'll say a Leber's patient and a glaucoma patient, is that you won't see as much cupping in the Leber's. The nerve tends to go pale, whereas in glaucoma you get pallor and you get this cupping as well, an altered structure on the surface of the nerve. So clinically you'll see a difference. But I think there will come a time when we will do, as we identify more genes for different forms of glaucoma and for their Leber's, we will probably be testing patients for the genetic defects associated with the different forms of hereditary optic neuropathy versus the glaucoma genes. That's coming, I think.
Okay, thanks. And just the last question there, just should everyone have an eye test irrespective of their vision, as this would detect glaucoma a lot more easily in the general population? True.
If you look at if you look at when glaucoma presents, and there's some great pictures that I could show, it only starts to appear really after the age of 60 for the majority of patients. Now, if you have a strong family history, it can occur earlier, but for the majority of patients with glaucoma, you only start to see it after the age of 60. And most people by the age of 60 already attend an optician to get reading glasses. Many have attended all their lives for glasses for short-sighted or long-sighted, but nearly everybody by the age of 55 is wearing reading glasses. So that's when you get tested. So that will help. It's fantastic that the opticians are in a position to look for the signs of glaucoma, and they now have the instruments to measure and monitor as well. So that's coming. That's a great help to us that everybody, is getting tested. But I'm not sure that everybody at the age of 20 or 25 needs to be checked unless you have a family history.
Okay, great. Thanks again, Colm. I think we move along for time, and thanks for joining us today. It was great to have you. Moving on to our final speaker of this evening is Dr. Alison Reynolds from University College Dublin. And Alison is going to talk to us a little bit about Charles Bonnet Syndrome. Some people might recognize Alison from, she's spoken at a couple of Fighting Blindness events. So it's great to have her back, and I'll hand it over to you, Alison. Thank you.
Thanks a million. So I'm just going to share, this is going to be very short. So in case any of you are like saying, oh, how long is she going to be? Don't worry, I'm going to try and keep it to about 5 minutes, okay? Hold on one second. Okay, so I wanted to talk to you today. Everyone can hear me and see the slides, I think. Do you just want to confirm that?
Yeah, we can see them, Alison.
Thanks, Rachel. Okay, so I just wanted to talk to you very briefly about a survey which I have been doing with Charles Bonnet Syndrome. And by me, I mean the royal we, and is of course my wonderful student Claire. I have a picture of Claire here in the slide. Claire has just gone on to do, she's now actually in the Mater doing her clinical placement at the moment, so she's a med student. So what we wanted to do was we wanted to learn more about a condition called Charles Bonnet Syndrome. And you may have actually received a survey from us. You may have received a little bit of information from us as well, or you may have heard Claire talking actually at the Patient Engagement Day last November either. So I'm just going to go on, and just before I do, I just want to say, I realize that we owe a debt of gratitude to Fighting Blindness and to many of the kind of, patients who have also participated in helping us design the survey as well. So if you're one of those, just thank you very much for helping us along the way as well. So what I should say at the outset is that we are still looking for people to complete this survey. We would like to know about the incidence of Charles Bonnet syndrome in the Irish population. And what do we mean by that is we would like to know how many people have Charles Bonnet syndrome. Now, this is a syndrome that only affects people who are, well, mostly only affects people who are visually impaired. And what it is it's kind of like a phantom vision. So people are seeing things that are not there. It was actually first described in about 1760. So it was first described a really long time ago by a philosopher called Charles Bonnet, who actually his grandfather had this condition, and he wrote it down, all of the symptoms of it. It's described in the literature, if you read it, normally as being rare. So as being rare, so in a kind of not really occurring much in a visually impaired population. But from talking to people here and from, I suppose, more recent evidence, we actually think it's quite common in people who have a significant amount of visual impairment. And essentially what people are doing is, like I said, they're seeing things that are not there. And it can be, I think, quite a different experience as well. And one of the things that we wanted to do was to raise awareness among visually impaired population as well, because I think it would appear to us kind of from our initial results of our survey, this isn't really talked about, and people have sometimes put it down to being kind of maybe worried about them having a mental health condition, which is not the case. So Charles Bonnet syndrome, I just have a couple of images on this slide, which there's a society in the United Kingdom called Esme's Umbrella, and they got people to kind of describe their experiences of Charles Bonnet syndrome. And what it is that people may see people who are not there. They may see kind of geometric shapes that are not there. They may see objects that are not there. And it might kind of seem, okay, well, that's just an interesting kind of a phenomenon for them to experience. But we do have concerns that maybe, say, if you were navigating your way down a road and there was a wall that wasn't there, or you saw a vehicle that wasn't there, it could impact your life in that way as well. And just to let you know, so we've had about 50 people complete the survey. So essentially we have kind of some preliminary results. All those people were adults, with visual impairments and living on the island of Ireland. So from this, we saw about half of these people had experienced Charles Bonnet syndrome. So you can see from that, it's not really very rare condition then. And the experiences of the visions really varied. So one of the things we were trying to get an idea about was kind of, what do you as a patient experience from this as well? So just the visions varied in terms of, some the types of visions that people saw, the frequency that they saw, and also then, I suppose, the length that they had the visions for as well. Now, in some people had a variety of different emotions towards it. Some people felt that were disruptive to their daily life. Some people, they said they were annoying. Some people said they were just kind of benign, so they didn't really bother them in the slightest. They did say they were impacted by stress and by tiredness, and the more vision you've lost, the more likely you are to have these as well. One thing we were concerned about, but it doesn't seem to be borne out, was did COVID, so isolation maybe due to COVID, make it worse, but it didn't seem to, we haven't really got any evidence of that at the moment. So the last thing that we kind of wanted to know was how people had learned about it and where they'd learned about it. And what we really say is that awareness is key. So this is really kind of like a bit of a hidden condition, maybe. A lot of people are experiencing it, and they kind of all seem to be experiencing it maybe in isolation and not talking to one another about it. So before the survey, 44% of the people who completed had not heard about Charles Bonnet Syndrome. And some of those were people who actually had Charles Bonnet Syndrome as well. So the positive thing about this is we've talked to the clinicians, particularly in the Mater and in the Eye and Ear Hospital, and we're hoping to raise awareness so that maybe when you turn up for your first ophthalmology appointment, that you will get some information about this as well. So it's not going to be something, you're not going to be sitting there worried that you have a mental health condition and you realize it's just a side effect of visual loss. Now, finally, I just wanted to say to you, the survey is available. Neve has sent it around, I think, in the most recent newsletter, or it's going to be in the next newsletter anyhow. And just to encourage anybody to complete it, anyone who's visually impaired, I should say. So one of the things we're trying to do is work out what numbers of people actually have experienced Charles Bonnet Syndrome. It won't be everybody. So even if you haven't experienced it, we're keen to hear from you as well. There's more information if you want to learn a little bit more on the Fighting Blindness website. And if you're really concerned, you could contact Fighting Blindness. They obviously have a counselling service. I think I have the wrong email address there. And lastly, if you want to, there's also information for our website, which is Esme's Umbrella. That's a charity in the United Kingdom as well. And it's charlesbonnetsyndrome.uk. And finally, I just want to say thank you to everybody who's helped us along the way. So to Fighting Blindness, to all of the patient advocacy groups who have helped us kind of, with finding out more information about Charles Bonnet Syndrome, with reading our surveys, with, helping us kind of have appropriate questions, and also then to clinicians as well. So in the Mater, in the Eye and Ear, and up in Belfast as well, who helped us design this as well. And as I said, Fighting Blindness. So just to say thank you very much. And if you get a chance, we really would be interested to hear your opinion on this as well. And that's me done. So I hope that was short.
Great. Thanks a million, Alison. Just a question that comes in a couple of times is it just visual hallucinations associated with Charles Bonnet, or?
Yes, so this is kind of what they say. Yes, normally it should be just visual hallucinations for Charles Bonnet. I know some people have, and that's actually one of the questions is, some people have reported maybe auditory hallucinations, or I think other sensations, like so it being able to smell things maybe that aren't there as well. But for Charles Bonnet, it is meant to be just the visual hallucination. Yes.
Thank you. Yeah, thanks very much, Alison. And I will be following up with people, and I will just include the link to the survey for anyone that mightn't have completed it yet. And that would be great if you could do so. Yeah, I think we don't have any more questions that came in. So thanks again, Alison. It's great to have you, and thanks to Claire as well for her work on the project. So we're nearly at the end of our session for today, and we just have a couple of minutes to share a video that's from Jackie Turner, who is a genetic counsellor based in the Mater Hospital. So Rachel's going to share that video a couple of minutes. Thank you very much. Cheers, Rachel.
Hello, I'm Jacqueline Turner. I'm a genetic counsellor in the newly formed Clinical Genetic Centre for Ophthalmology in the Mater Hospital. The unit was developed in response to a need for a genetic counselling service to return genetic test results to patients and their families who were tested by the Target 5000 team at the Mater. The Target 5000 study began in response to a new emerging approach in medicine called precision medicine. Precision medicine allows treatments to be selected based on the genetic factors underlying a patient's disease. Gene therapies, where you are putting a working copy of a gene back into the eyes of patients that are missing that gene, is an example of precision medicine. All patients participating in the Target 5000 study have samples taken for genetic analysis. This is performed first in the Trinity Research Laboratory, and there they find the spelling mistake, which they believe is causing your eye disease. Once we have that, we can then send your sample over to our diagnostic laboratory where they can confirm the result. The diagnostic lab will also produce a genetic report. We then meet as a team and to determine if the patient's photos and eye examinations and family history fit with what has been found in the genetics report. We also check that the changes found are classed as disease-causing. This is very important as we have millions of changes in our genetic material, most of which are harmless, some are beneficial, and only some are disease-causing, and we need to be confident that the changes found are causing your condition. Genetic counselling involves helping patients understand the cause of their eye condition and how it is causing their visual impairment. I'll also discuss how the eye disease is inherited and what are the chances of a patient's children developing the condition, and who else in the family could be at risk of developing the condition. Once we are confident that the changes found is disease-causing, we can offer testing to those who are at risk, if they want to know, to determine if they are likely to develop the condition in the future. We are at a very exciting time in medicine with therapies that are emerging that can halt the progress and even improve the vision of those with genetic eye disease. And we hope in the future that these therapies may work at curing eye disease. We all have genes which make proteins which direct our body's processes and ensure that our body is working and functioning normally. The new genetic testing allows us to examine many genes at the same time. There are over 300 genes which we need for our retina to work and function normally. With the new genetic testing methods, we can screen all these genes together to see if we can find a mistake in one of the genes which is causing our visual problems. We can then check that that mistake is actually disease-causing. Often the changes found in the genes are unique to the family or haven't been described yet. We are very new to this. The human genome was only first sequenced in 2003. By finding the gene involved, we can learn more about how a particular gene is causing the condition. For example, you may have been diagnosed with retinitis pigmentosa, but there are over 80 genes which can cause retinitis pigmentosa. And perhaps some genes cause a late onset type. Other genes might cause an early onset type. Some genes might cause a slowly progressive type. And for other genes, the progression might be quite quick. So in order to know how a particular gene is causing your condition, it would help us in the future with your prognosis. Knowing the gene helps us to determine how the condition is also inherited. Having a visual impairment affects people differently. Everyone who develops a visual impairment throughout their life will grieve their missing vision in some way. It is quite normal to feel sad that you do not see as well as you used to. The important point here is that you're not alone. There is a team of people available to support you and to try and enable you to accept your loss and to move on with your life, concentrating more on what you can do rather than what you can't, so that you can live your life to the full. It is important to remember to do what you enjoy, and if you really can't do that, to investigate other interests and hobbies that you might enjoy. There is also much more support available than there was in the past in education and in the workplace. And technology has enabled those with visual impairments to participate as fully as possible in what is going on around them. There are so many gadgets and apps available to assist you. There is always a solution. If you are part of the Target 5000 study and you wish to be seen for genetic counselling, please ask your clinician to refer you to me. Thank you.
Okay, I'm just going to hand over to Rachel, who is the research manager of Fighting Blindness, to close the session, but just personally to thank all the speakers again today for giving their time, and they were all really excellent. So
thanks, Niamh. Yeah, just to wrap up and thank everybody. Thank you so much to our speakers. I know I found it really informative. I'm quite new to the role and I learned a lot. So thank you very much for giving your time this evening. Thank you to our sponsor, Roche, who are sponsoring this webinar this evening. Most importantly, thank you to everyone that has attended this evening. We really appreciate it. So as Niamh said, she'll be in touch with a link to the survey that Alison spoke about, but also a link to the recording of this webinar. And we'll be sending out a short survey just to get some feedback, because we're always looking to improve and get ideas from people. Also, our contact details are on our website, but if you want to email the research team, it's best to email research@fightingblindness.ie, and we can get back to you on any questions that you might have after this webinar. So yeah, that's everything, and thanks again to everybody, and I hope everyone has a lovely evening.